So let's know about a plant hormone known as brassinosteroid. They are the class of plant hormone which are essential for various physiological process including growth, development and stress response. These are the steroidal compound found in the plant and are analog to animal steroid hormone in their structure and function. So, the key features of this brassinosteroids are growth regulation, stress tolerance, developmental process, hormonal interaction, molecular mechanism. These are some of the key features of the brassinosteroid. So, first we will know what is this growth regulation. It stimulates the cell elongation and division. It promotes seed germination and root growth, and it enhances vascular differentiation. If we see in stress tolerance, it helps the plant to cope up with environmental stress like drought, salinity, extreme temperature, and heavy metals. It regulates antioxidant system in plants. In developmental process, it influences flowering and fruiting development, and it regulates leaf senescence and photomorphogenesis, that is light dependent growth. If we see hormonal interaction, it interact with other plant hormone like auxin, gibberellin, cytokinin to modulate plant growth and development. In molecular mechanisms also, brassinosteroid bind to the specific receptor like brassinosteroid insensitive 1, that is BRI1, on the cell membrane, and this triggers a signaling cascade that lead to change in gene expression impacting the growth and stress response. For example, brassinoloids, the first brassinoid discovered, testosterone, a 24-epi brassinoloid naturally occurring form. If we see the applications of this brassinosteroid, it is used in agriculture and biotechnology. In agriculture, they are used to enhance crop yield and improving resistance to pests and diseases. In biotechnology, research focus on manipulating BR pathway to engineer stress resistance and higher yield of crops. If we see the biosynthesis of brassinosteroid, they are synthesized through a complex pathway in plant derived from sterol precursor like campestrol. This process involved multiple enzymatic steps and pathway including earlier C6 oxidation pathway, the major route of brassinosteroid biosynthesis, and later the C6 oxidation pathway, an alternative route for brassinosteroid production. So the key enzyme involved in this are catalyze DWAR F4. This catalyze hydroxylation step critical for brassinosteroid activity and constructive photomorphogenesis and dwarfism, that is CPD, involved in hydroxylation of intermediates, and then DET2, that is the de-ethylated 2. It catalyzes an earlier step in brassinosteroid biosynthesis similar to mammalian sterogenesis enzyme. If we see the mechanism of action in brassinosteroid signaling pathway, it involves perception. So brassinosteroid bind to a cell surface receptor kinase, that is BR1 insensitive 1, brassinosteroid insensitive 1, and its co-receptor BAC1, that is BRI1 associated kinase 1. The signaling action in it, binding activate BR1 and trigger a phosphorylation cascade, and it deactivates BIN2, that is brassinosteroid insensitive 2, a negative regulator. After that, what happens? The gene expression regulation happens. The deactivated of BIN2 allow transcription factor like BES1 and BRI1 EMS suppressor 1 and BZR1, that is brassinosole resistance 1, to move into the nucleus, and these transcription factor then regulate brassinosteroid responsive gene and influencing the growth and stress response. Now let's see the function of brassinosteroid. In growth and development, it promotes cell expansion, cell division by altering cell wall properties, and it regulates stem elongation and root development. In flowering and reproduction also, development, it enhances the pollen tube growth and seed development. If we see the stress response, it improves the tolerance to abiotic stress such as drought. It enhances water use efficiently, and in salt stress, it modulates the ion transport to maintain the ionic balance. If we see the cold and heat stress, it regulates the antioxidant enzymatic activities to reduce oxidative damage. If we see the leaf senescence, it delay aging by suppressing senescence related gene, and it enhance photosynthetic efficiency. Stress mitigation. Foliar spray of brassinosteroid are used to help crop tolerance, drought, salinity and heat. If we see pest and disease resistance, it enhances the plant initiate defense mechanism against pathogen and insect. If we see the post harvest improvement, it delay fruit ripening and senescence and maintain the quality during the storage. Some examples of brassinosteroid, they are natural brassinosteroid are brassinoids. They are the most active and widely studied BR, and the castasterone, an immediate precursor to brassinoid. So 24 epibrassinoids, a potential natural BR, developed for commercial use and growth promotion. The signaling pathway is well studied mechanism that govern plant growth and development. It involves a series of interaction for cell membrane to nucleus, modulating gene expression. We have seen what is BR1, BRI1, that is biosynthesis insensitive 1, the receptor and co-receptor in cell membrane. This BRI1 is a leucine rich repair receptor like kinase. The LRRRLK located on the plasma membrane. It binds to the brassinosteroid receptor, initiating the signaling cascade. Then BAC1, this is BRI1 BRI1 associated kinase 1, a co-receptor that forms a complex with BRI1 upon BR binding at enhanced BRI1 activation, then signaling protein in the cytoplasm. So BSK, that is BR signaling kinase, phosphorylated by BRI1, BSK transmit the signaling to downstream, and BIN2, that is brassinosteroid insensitive to GSK3 like kinase that act as a negative regulator of BR signaling, and in the absence of BR bin2 phosphorylate transcription factor preventing their activities. After that, the protein phosphorylate 2A, a phosphate that deactivate bin2 facilitate BR signaling, and then transcription factor that is inside the nucleus. So BZR1, that is brassino zole resistance 1, and BES1, that is BRI1 EMS suppressor 1. These are the key transcription factor that activate in response to BR signaling, and they regulate BR responsive gene involved in the growth and stress response. So next step in brassinosteroid signaling pathway is BR binding to the receptor. So what happened? The BR molecule bind to the extracellular domain of BRI1, and it cause a conformational change, and this activates the BRI1 and recruits the co-receptor BAC1. So the receptor activation, that is BRI1 and BAC1 complex, undergo autophosphorylation and phosphorylates downstream signaling protein including BSK and CDG1, that is constitutive differentiational growth 1, and inactivation of BIN2 takes place, and then the activated BSK and CDG 1 phosphorylate and activate BSU1, that is BRI1 suppressor 1. Then BSU1 dephosphorylate and inactivate BIN2, a negative regulator of the pathway. Then activation of transcription factor take place. The deactivation of BIN2 allow BZR1 and BES1 to become dephosphorylated by PP2A, that is dephosphorylated BZR1 and BES1 enter the nucleus to regulate BR responsive gene expression, and this gene expression changes occur. Here the BZR1 and BES1 bind to the BR responsive element, that is BRRE, in the promoter region of the target gene. These gene control process like cell elongation, cell division and stress response. The summary of this pathway without BR is BIN2 is active and phosphorylated, and BZR1 and BES1 keeping them inactivated in the cytoplasm, and with BR, that is with brassinosteroid, the brassinosteroid inactivate Bin2, allowing BZR1 and BES1 to move into the nucleus and activate target gene. If we see the diagrammatic representation of this, the cell inside the cell membrane, Br getting converted to BRI1 plus BAC1, it gives to signaling. And then if we see the cytoplasm, signals are then inactivate the bin to then activate BZR1 and BES1. In nucleus, BZR1 and BES1, then activation of BR responsive gene occurs. The key gene regulated by BR signaling are growth related gene, that is cell elongation and cell division. Stress responsive gene are antioxidant enzyme regulation, and developmental gene are flowering, seed germination and root development. Plant parts do we get brassinosteroid? They are in the young leaves. The high level of brassinosteroid are found in the developing leaves, as these hormone promote cell elongation and cell division. If we see the shoot apices, the brassinosteroid are concentrated in shoot meristem, where they regulate growth and morphogenesis. If we see the reproductive structure, the pollen and anther, the brassinosteroid are abundant in the pollen grain and anther, where they facilitate pollen tube growth and fertility. If we see in the flower also, they are found in the petal and floral bud, supporting flower development. In seeds and fruits also, immature seed, brassinosteroid play a role in seed germination and earlier growth process. If we see the fruits, then brassinosteroid are present in developing fruits, where they regulate the size, shape and ripening. If we see the roots, they are present in the root tip, where they influence root elongation and branching. However, the concentration is generally lower than in shoot. If we see the vascular tissue, it is found in xylem and phloem tissue, contributing to vascular differentiation and transport efficiency. Parts of the plant such as cotyledon in higher level in young cotyledon, aiding in earlier seedling development, and in internodes also it prominent in elongating internodes, promoting stem growth. If we see the variation in brassinosteroid levels, then highest concentration found in reproductive structure, for example pollen, seed and flower, and young actively growing tissues, for example shoot apices and young leaves. In lower concentration also it is detected in mature leaves, roots and older tissue, as their growth promoting role diminish. Brassinosteroid, they are a naturally occurring plant hormone, and they are found in many wide ranges of plant families, and they are involved in growth and development. These brassinosteroid are ubiquitinated across the plant family, and some plant family and species are notable for their contributions to brassinosteroid studies, particularly in the term of discovery and analysis of these hormones. So the plant families producing brassinosteroid are mustard family and or Arabidopsis thaliana. Some importance of this particular, the first brassinosteroid was discovered in Brassica napis pollen, and Arabidopsis thaliana is widely used in this research for studying biosynthesis and signaling pathway. Then the second family, grass family, Poaceae family, for example rice, maize, barley, wheat, all are containing this particular brassinosteroid. So Poaceae member are significant for agriculture research, and brassinosteroid is in these crops enhance grain yield and stress tolerance. If we see Solanaceae family, that is the nightshade family, for example in tomato, potato and tobacco, its importance is used and regulated in fruit development and stress response in economically important for crop like tomato and potato. If we see Fabaceae family, in legume family also, soya bean, pea, alfalfa, these are some of the plants in which it is formed. Its importance is critical for seed development and symbiotic nitrogen fixation in legumes. If we see in Asteraceae family, sunflower family, in it also it has some importance. For example, it have contributed to understanding their roles in stress tolerance and growth regulation. If we see the gourd family, that is Cucurbitaceae family, in this pumpkin, cucumber, importance of this particular hormone are linked in the fruit elongation and resistance to environmental stress. If we see the citrus family, that is Rutaceae Um, in that, orange and lemon are an examples of this for this hormone, and they are associated with fruit size and ripening of regulations. If we see Euphorbiaceae family, in it, rubber tree, castron bean, these are the some of the examples. In its, it plays a role in stress adaptation and latex production. If we see the mint family, Lamiaceae family, for example, tulsi, that is basil ocimum, and mint plant also, it is important the growth and essential oil production in aromatic plants. In Malvaceae, mallow family, such as cotton and okra, some important role in brassinosteroid is that it promotes the fiber elongation in cotton and improve the yield under stress. So they are found in angiospermic flowering plants as well as the gymnosperm, making them nearly universal across the plant families. And plant families with significant economic importance, for example, Poaceae, Solanaceae and Fabaceae, have been extensively studied for this particular hormone.